So previously I described ideas. Basically, so far I haven't explained methodology in details, but describe just concepts, main concepts, and main ideas, how you can see omics integration through machine learning lenses. So basically, I think what I would like you to learn is that prediction is absolutely central. You measure success of your omics integration through prediction. Um, and you have to do, unfortunately, very often feature pre-selection in order to overcome the curse of dimensionality. The curse of dimensionality is maybe one of the major challenges when you combine multiple data types measured for the same biological samples, and you need to handle it somehow. Otherwise, um, very often you get meaningless results from R or Python packages that simply formally can deliver something, but you can never be sure that it's a correct quantification. So now I will explain how you do feature pre-selection and concentrate on supervised omics integration. So again, this table that I briefly showed you on one of the last slides from the previous lecture, it sort of structures different integrative methods. So when you start with an integrative project, you should ask yourself whether I have enough samples to do non-linear integration. This is maybe affordable when you work with single cell data where you have hundreds of thousands of cells or maybe even millions of cells. This is not uncommon. But I think for majority of biological and biomedical computational biology projects, we are still sitting in the linear limit. We can't go to neural networks or random forest machine learning methods. It's a little bit risky. You often, if you just apply AI to underpowered study design, you're at risk to get something not generalizable, something that will be working here now for your particular data. But if you take your model and go to another lab and try to do some integration or prediction with this model using data from a different lab, you'll most likely fail. This is what is called overfitting, a lack of generalizability of your model. And then if you sort of, sometimes you have a clear idea, it can be some clear disease or clear phenotype of your cells. You have maybe good cells and bad cells. Then, of course, you can always do unsupervised analysis. But if you have a clear interest, if you want to learn something about your good cells and bad cells, or sick individuals and healthy individuals, you should better do supervised analysis. But otherwise, even if you have a clear phenotype of interest, maybe diabetics and non-diabetics, but still you can ask, maybe there are subtypes of diabetes or non-diabetics can be also subdivided into subgroups. There is some hidden structure in the data. And then unsupervised analysis is very useful in this case. But today, so now, right now in this lecture, I will be mostly talking about these models, this type of analysis, linear and supervised, because this is sort of, to my experience at least, it's quite common. So again, I briefly jumped over this slide, so here I will sort of again reiterate the protocol that I suggest. It's not written in stone. You might disagree. You might have better ideas. So please let me know if you sort of have better suggestions. But this is how I sort of see applying machine learning to multiple data types in computational biology. When you have multiple data types, maybe you have gene expression, methylation data, genotype, and phenotypic data. And you have a clear phenotype of interest, diabetes, maybe, as an example. And you, out of your 110 samples mentioned here, you have 29% of diabetics. So roughly 30% of the samples are cases, the rest are controls. So the first step you do in any machine learning, you always split your data into train and test data set. Sometimes you have batches, and this is actually beneficial for machine learning. People tend to correct for batch effects and say batch effect is something bad, we want to get rid of this. But for machine learning, it's actually good to have batches because in machine learning mentality is not based on merging data types. Because if you have a batch, smaller batch, you can use it as a test sample. So batch effect is a natural split of your data into train and test. So you don't need to actually merge your different batches, but use one of them, for example, as a test data set for validating or evaluating your model. So once you split your data set into train and test subsets, you need to proceed with feature pre-selection. So if you have a lot of samples, you might want to skip this step at all. If you can afford using all features. Of course, ideally, we would like to use all features and not to pre-select them. But unfortunately, often in practice, we can't do it because of the curse of dimensionality and all these mathematical pitfalls that I described previously. And there are sort of two different ways of doing feature pre-selection. So if you have a clear phenotype of interest, such as type 2 diabetes in this example, you can use, for example, Lasso. It's a very popular, famous method for extracting most informative features, genes, for example. You assume that there is some redundancy in gene expression matrix, that not all genes are independent. And we know that many of them are not independent. They do co-express. So you want to, there is no point to keep all of them because this contaminates your analysis. This brings redundancy into your analysis and mathematical difficulties with divergences that I explained previously. So you want to get rid of the noisy genes, lowly expressed genes, for example, and noisy genes. This can be done through applying Lasso, but Lasso is a supervised approach. So you have to provide a response variable. You have your genes as explanatory variable, and you have your response, which is, for example, type 2 diabetes. And you select only the genes that already have some sort of relation to your phenotype of interest. Otherwise, if you want to be sort of open-minded and don't want to use this phenotype of interest and don't want to narrow down your set of genes to the set that is already related to diabetes, you can use unsupervised feature selection. What you do here, you basically look at some measure of variation of your gene expression. For example, if you are talking about gene expression data, you look at how much each of your genes varies across samples, and you select only the highly variable genes. because why you select highly variable genes? Because you assume that there is some difference in gene expression for some genes between cases and controls. If there is no difference in gene expression, if you take the gene and there is no difference in gene expression between cases and controls, there is no point to keep this gene for your analysis. It's basically non-informative gene, but you will contaminate your analysis. You bring redundancy to your analysis if you keep this gene. And then you do integration, so in supervised fashion, and using one of these methods. And once you train your model, you can bring your trained model and evaluate it on the test data set. And ideally, you should do it a number of times. So you do this split into train and test a number of times, maybe 1,000 times. Why you want to do it? You want to build confidence intervals. So you know that a measure of uncertainty is always important when you do predictions. If I predict that a certain individual is based on molecular data, 80% chance that this individual is diabetic, then it's better I have a confidence interval around this 80%. If the confidence interval is huge, maybe it can be 200%. Imagine that I predict that this individual is diabetic with probability 80% plus minus 200%. Then it's probably not a good prediction. So this uncertainty is a crucial measure when you do prediction in, for example, for clinical diagnostics. It's very important to understand the uncertainty of your prediction. So that's why you want to do this split into train and test set in order to make sure that your evaluation of the model is not the result of just lucky split. You need to do the split multiple times and make sure that and that you also provide an uncertainty estimate when you do prediction. So I hope it's a little bit more clear now. Yes, so here I sort of remind you that there are notoriously difficult data sets. For example, if you work with genetic variation using whole genome sequencing data, then you end up with millions of genetic variants, or if you use whole genome bisulfite sequencing, you also end up with millions of CpG sites. Even if you apply feature pre-selection, if you try to reduce dimensions of this individual omics somehow, it's still too much. And typically, the concepts that I'm describing, they typically will not work well if you have that high dimensional data. So if you have ultra high dimensional data, unfortunately, you will have to increase your sample size somehow. Otherwise, it's meaningless basically to continue with analysis. Another reason why you want to do feature selection is again, it's all again boiled down to the disadvantages of maximum likelihood approach. It's commonly used. However, one needs to remember that this maximum likelihood approach, it doesn't really work when you have some sort of non-independence in your data. So maximum likelihood or Gaussian distribution assumes that all your data points are independent. If there is some clustering between your data points, then this assumption of independence, it's violated. And you need to account for this somehow. For example, you can account for non-independence between your samples using linear mixed model, and account this clustering through random effects. That's how you make your assumptions fulfilled by introducing a separate term. into your, for example, linear regression model. Or, so this was a non-independence between samples. But you can also have non-independence between features. So again, remember that in your data matrix, you always have samples and features. And you can have sort of non-independence both between samples and between features. And if you have non-independence between features, such as co-expression of genes in RNA-seq data, you better not to include all of them, not only because it's just silly to include redundant genes, but also because the more non-independence you introduce into your data, the more you violate the very fundamental assumptions of the maximum likelihood principle. And then you better do feature selection with Lasso, for example, to make sure. So Lasso basically, ideally, brings you a set of features that are not correlated. Yes, so that's another reason for doing feature pre-selection. But it's all related to the curse of dimensionality. So this motivation is also can be explained in terms of the curse of dimensionality. And here I will talk about univariate and multivariate feature selection. So there are two types of feature selection. A very simple feature selection that you can do, and you will be doing it in the exercises. So I simply explain it here using gene expression data from skeletal muscles from GTX consortium. There is a big consortium, GTX, with gene expression data from human individuals from, I think, over 70 different tissues. And for example, if you take skeletal muscles for 157 individuals, so, and I plot, I make a PCA here, a simple PCA demonstrating different samples. So one point here is one sample, and circles are females, and triangles are males. And for example, I want to, who knows, maybe I'm interested in gender as my main phenotype of interest. So unfortunately, GTX does not provide many phenotypes. That's why I chose gender as a very simple phenotype. And in your case, it can be a disease, for example, or some morphology of a cell. But now what I want to select genes that are somehow related to gender. So I randomly sampled 1,000 genes, and I am asking a simple question. Can I pre-select my genes? Can I choose? I don't want to have 1,000 genes. It's still too much for me. I want to have maybe 10 genes. I want to reduce dimensions of my data set. I want to pre-select features. And one simple thing you can do, you can just do differential gene expression, for example, or like a correlation, like what I'm doing here. I'm correlating each gene. So I simply correlate each gene with gender. And I'm saying, I want to rank all my genes by their correlation with gender. So there will be a lot of genes, maybe coming from X and Y chromosomes, that are strongly correlated with gender. And they will be on the top of this list. And now I say, okay, maybe I want to now take maybe 5% of most strongly correlated genes, strongly correlated with my phenotype of interest, which is gender. And in this way, I've done feature pre-selection. So I reduced dimensionality of gene expression. However, I've done it in so-called univariate fashion. So what I was doing, I was testing gene by gene. I did not test all of them simultaneously. This is what is called univariate feature selection. That when you test feature by feature, you simply do statistical test, and then you don't consider relationships between genes, but you test them individually. And later I will show you a slide comparing comparing predictive power of univariate and multivariate feature selection. It's pretty clear, at least from my experience and what I read, that basically doing feature pre-selection in univariate fashion is not an optimal way. So multivariate analysis, the reason why people very often use multivariate analysis is simply because it's a more robust and more robust in terms of prediction type of analysis. Multivariate analysis generally has a higher predictive capacity compared to univariate analysis. And one of the famous tools, if you want to do instead of differential gene expression, if you want to do multivariate feature selection, you can do it, for example, with Lasso. And what Lasso does, so imagine that Y is your phenotype of interest, maybe type 2 diabetes, and X, or gender, let's stick to gender in my case, in the previous example. And X1 and X2 are two genes. So what you do when you're solving this linear regression model, so epsilon is an error, that's your model, a model has an error. What you typically do when you solve this linear regression equation, you minimize the error epsilon, and it's called ordinary least square approach. So by minimizing the error between your model and your true labels, males and females are true labels, you minimize the error between your true labels and your prediction. So on the right-hand side, there is a prediction of your model. And if you compare now this prediction with the true labels, that will be the error. And you want to minimize this error. And this is a so-called ordinary least square framework. So what Lasso does in addition to that, Lasso adds a penalty in this very strange way. So you add some coefficient lambda. I'll explain what it means. Multiplied by absolute values, a sum of absolute values of beta 1 and beta 2 coefficients. And then you use a procedure which is called cross-validation. And in a second, I will explain what it does. You use this cross-validation procedure in order to find an optimal value for lambda. Lambda for now is just an abstract coefficient. In fact, what Lasso is doing, what is shown here, is a very well-established method in mathematics of optimization, when you include boundary conditions with so-called Lagrange multiplier. So what is shown here is so-called Lagrange multiplier approach for functional optimization. And this lambda, optimal lambda, a value for lambda, can be found through monitoring the error of your model as a function of possible values of lambda. And usually this error of your model has a minimum at certain lambda, and this will be the optimal value. And so now, how do you compute the error of your model? You compute it through, as I said, a cross-validation procedure. And here I explain what is k-fold cross-validation. There are other types of cross-validation, but for simplicity, this is what I meant by a k-fold cross-validation. So you simply split, again, your data set. For example, if you use 5-fold cross-validation, you split your data set into 5 different subsets. And you use 4 of them for training your model, and the last one for validation. Validation means that you use your trained model and you do a prediction on the test data set. And then you compare your predicted labels. For example, I predict whether my sample is male or female based on gene expression. So I don't know the truth, but I predict that based on gene expression, this sample looks like a male. But then I can compare my prediction with a true label and compute an error. My model will be a little bit wrong always, but I can minimize this error. So you compute the error once, And then you choose, you do another split, you do another iteration. You, again, use this blue segments for training and the white one for validation, and compute error again, etc., etc. You do it multiple times, and you compute sort of a collective error of cross-validation. This is what is plotted here. So y-axis here displays the error of your model through cross-validation. Why you do this cross-validation? You typically cross-validation is a standard way to tune hyperparameters of a model. So a model always have parameters and hyperparameters. So if you look at this penalized OLS, penalized ordinary least square. So now we are talking about a family of models that are called penalized linear models, because you add a penalty here. So this model has parameters where beta 1 and beta 2, those are parameters of the model. While lambda, this is a hyperparameter of the model. So it's sort of a parameter that is more kind of general for all types of parameters. So basically all models typically have parameters and hyperparameters. Sometimes it's difficult to figure out what is a hyperparameter. But in this case, for Lasso, it's very obvious. Maybe if you do machine learning, after some time, it will be very easy for you to look at any model and immediately see, okay, this is neural network. Okay, these are parameters. The weights of the edges in my graph are parameters, and the number of layers is a hyperparameter, for example. Yes, and typically you optimize hyperparameters of any model through cross-validation. Yes, and it's not only Lasso that does this penalized regression trick, but there are also, maybe you heard, there is a Reach and ElasticNet, different models. They all are different in terms of the penalty you add. For the case of Reach, it will be quadratic, beta 1 squared plus beta 2 squared. And for the case of ElasticNet, it will be some combination of basically of this penalty and Reach penalty, quadratic penalty. But they're all about the same. However, the difference between them, roughly, is that Lasso is more conservative. Lasso is the most conservative out of them. And it means that basically the idea of adding a penalty to your model is to force your model here, to force your model to prioritize either beta 1 or beta 2. So basically, by adding some positive constant, some positive contribution to your objective function. So this is an objective function, and you add some positive contribution, and the model wants to minimize, to make this error, this objective function, which is an error, the model wants to make it as small as possible. But you add something, some penalty, some positive contribution. So you increase your error. And in order to survive this optimization procedure, the model has to prioritize to keep either B1 or B2, or sorry, beta 1 or beta 2. That's why you prune your set of genes. You select only the ones that are most informative, the ones that have largest coefficients, that have strongest correlation with your phenotype of interest. And basically, Lasso does it in a most conservative way. So you end up with the smallest set of features if you apply Lasso. And this is actually very good for computational biology, because biological data types, they're very noisy. You really have a lot of technical factors, and you better be as conservative as possible if you want to have a generalizable model. So basically, compared to Reach and ElasticNet, Lasso is maybe sort of delivers you as few features, as few genes as possible but those will be for sure the most informative ones. Here I simply compare predictive capacity of Lasso, Reach, and Elastic Net. And I think it's pretty clear that for this gene expression from skeletal muscle data set and using gender as a response variable, I visualize the predictive capacity of these three models through so-called ROC curve. It's a standard way to measure sensitivity and specificity of the model. And Lasso sort of, and Elastic Net in this case for this data type, they kind of seem very different in their predictive capacity compared to Reach. So Reach is very permissive. Reach will give you a lot of genes will survive Reach optimization, while only a few genes will survive Lasso optimizations, in brief. And another interpretation of Lasso model is that you kind of you can view this penalized regression through balance between bias and variance. So in machine learning, it's very famous and widely used concepts, bias and variance. So what it means is that essentially so your model has an error always, as I said, and you can show that the error can be decomposed into two positive contributions that are called bias and variance. They are sort of uncorrelated, bias and variance. And it turns out that in order to minimize error, it's not good enough just to ideally you would want to decrease both of them. You would like to have this kind of situation when you have low variance and low bias, then your predictions will be on target. But in real life, it's not as good as this. In real life, you often prioritize either bias or variance, depending on your purposes. For example, if you have low bias, and by the way, penalty, the penalty which Lasso introduced here can be seen as increasing the bias of the model. You bias your model deliberately in order to make it more generalizable, in order to decrease variance. So you sort of, yes, sorry, with Lasso, you're sitting here. This is your case. When you increase bias, when you add the Lasso penalty, you might be missing the target, but at least your model will be very generalizable. And this is generally, or often people want to have. They want the model work on this sample the same way as on a sample from another lab. Because if it doesn't, like here, the opposite scenario would be a low bias, but high variance. So sometimes you get very close to the target. And again, you don't know where the target is. You just see that your sort of predictions are very different between samples. You use one sample, you get this kind of prediction, then use a sample from another lab, you get this kind of prediction. So you have very, very huge variation between your prediction. And this is sort of not good either. So it's hard to say whether you should prioritize bias or variance. But what I want you to remember is that adding penalty, Lasso penalty, it means that you're increasing the bias or you reduce complexity of your model. So here, this plot shows the x-axis shows here the model complexity. So you make your regression simpler by excluding many genes. So your model becomes less complex, but you also decrease variance. So your model becomes generalizable. So what Lasso does essentially, it makes your model, your prediction, your machine learning framework more generalizable, which is often what people want to have. So now I move, so it's a few minutes left. So you will be doing exercises on one of supervised integrative omics method, which is called Diablo. It's a Diablo algorithm. So I finished with feature selection. So up to now, it was a feature selection. And now a few words about Diablo, about the integrative method itself. So you pre-selected features, and now you proceed with integrative method. And it's better to learn Diablo by applying it. But here I briefly describe the idea. So what uh, basically Diablo is a PLS-based method. PLS stands for partial least square regression, or the author of PLS, actually Swedish author, called it projection to latent structures. But this is too difficult. It's a difficult name. That's why in English-speaking literature, people often say partial least square regression, PLS. And what PLS does, so simple idea behind PLS is that in contrast to PCA, so here I was showing a PCA plot for using gene expression as underlying data. But this PCA was done, so different colors, they display males and females. So you see the dramatic difference between PCA and PLS. Yes, here. You see that now the samples, males and females, they tend to be more separated. This is exactly what PLS does. It's sort of, you can think about PLS as taking a PCA and then extracting features on the X and Y axis in your principal components. Let's select features that provide the largest separation between my classes, between males and females in this case. So basically essentially we do feature selection through PLS that maximizes the separation between my classes in some latent space. And then you can also visualize loadings similar to what PCA delivers. It delivers interpretation of the model. It delivers loadings where all features, all genes are ranked by their importance. So how much each gene contributes to this separation between the classes. So this is yes, sorry, the Diablo that I was going to explain, it's based on PLS. So far it was not Diablo, but it's a multivariate feature selection through PLS. But it's in order to understand Diablo, you have to understand what PLS does. And then finally, there was one more method that I would like to mention for you, so that you know there is another interesting multivariate feature selection method, which is called linear discriminant analysis. And what it basically does, it tries to separate your sample. So now it's another data set, sorry about that. It's not males and females anymore, but something else. But what linear discriminant analysis does, it also tries to separate your samples and select appropriate features that separate my different classes. But it does it like technically through minimizing variance within clusters and maximizing variance between clusters. So if you think deeply about it, you can see a lot of similarity with PLS. Essentially, it's all about the same. So LDA, PLS, and Lasso, they are conceptually doing very similar things. That's why they can be used interchangeably, and they often result in a very similar sets of features. And here I also I promise to show some comparison of predictive power between univariate and multivariate methods. So I just, if you're interested, because I mentioned previously that you should do feature selection through multivariate methods. And usually when you do any type of analysis, you should think at least about multivariate analysis because it tends to have higher predictive capacity. And here I sort of compare a few multivariate and univariate methods. So it's hard to like immediately to understand what all these abbreviations mean. But briefly, sort of three top methods that have the largest area under the ROC curve are actually multivariate methods, Lasso, PLS, and random forest, while all other methods, they were univariate. And basically, I think it's a clear demonstration that at least for this skeletal muscle gene expression data set, multivariate methods, they outperform univariate methods in terms of prediction. Um, and finally, Diablo. So I explained already PLS, the concept of PLS. And Diablo, indeed, it uses PLS as a basic method. Diablo belongs to Mixomics R package. And Mixomics, it's actually not only Diablo, it includes other methods such as Mint, and this is integration across samples. So Diablo is integration across omics. I think I talked a little bit about the difference between integration across samples. I said what Mint is doing is actually similar to batch effect correction. Well, Diablo is doing integration across omics measured on the same biological samples. And basically what Diablo does, if you sort of try to digest this, at the first glance, it looks very complex, this equation, but I still sort of advise you to think about this equation because you understand in depth actually what Diablo is doing by looking at this. What essentially Diablo is doing, it computes a sum of pairwise covariance matrices between different omics and maximizes this covariance. And then sort of, in other words, you extract a part of variation that is common between your different data types and try to factor it, to split it into independent components in order to interpret your model. And here the coefficient, so here I and J indexes, they correspond to different omics, and C prefactor, it actually measures a prior knowledge about relation, a priori relation between your different omics. And this is a hyperparameter actually, while beta here is a parameter that you optimize. Yes, so you will be doing this exercise using chronic lymphocytic leukemia cell data, gene expression, mutations, methylation, and drug response. This data set, there will be a reference to the publication for this data set. And essentially what you will learn during this exercise is how to apply Diablo and get this kind of consensus plot. It's called error plot. When you project all your different blocks, so you have 4 blocks for data types, but then you you project all your different data types on a common latent space. And this picture is a result of integration. It's a result of sort of averaging across all your omics. And here in this latent space, you can clearly discriminate between females and males. While using individual omics, sometimes it's questionable whether you can actually achieve this. Only gene expression shows a clear difference, but other data types, this difference between males and females is less obvious. And yes, so for validating your model, so essentially what I've shown here, what any machine learning method does actually, it simply learns a boundary between different classes in some common latent space. So you project your data into some strange latent space where it's easy to just linearly separate your different classes. So this is a training. This is what training does. It draws the border between your classes. And then the field circles, those are actually the test samples. You validate your model by projecting now test samples on this trained model and checking whether my black field circles, they end up on the right or left side from the boundary. So this guy, for example, was misclassified because this part of the space was corresponding to red class and not the black class. So I hope this sort of gives you a little bit of intuition how you evaluate the error of your model. So you first train your model, and then you take test samples and see where they end up. If they end up, if the black guys, they usually end up on the left side from the boundary, then it's a good it's a generalizable model. If there is some misclassification, then you sort of it's a less accurate modeling. And Diablo, in addition, provides a lot of interesting visualization of your data. And everything in Diablo is done through cross-validation, which is a very nice, very robust procedure that you should learn and use it as much as possible, because this makes your computational analysis much more generalizable. And so I skip sort of other slides.
We can take this tomorrow, perhaps.
Yeah.
Since you will be talking about deep learning tomorrow.
Exactly. So yeah, I think feature selection and Diablo will be enough now. So you can continue with the exercise after the break. Yes, after the break, after the lunch. And I will be happy to talk more about Diablo and answer your questions, but there is a lot of explanation. When you will be doing the notebooks on Diablo, you will be learning all the steps in Diablo pipeline. And I think I try to make all the steps as clear as possible. So as I said, Diablo is better to sort of learn when implementing it to your multi-omic data set. So thank you very much.